Good afternoon. Sorry, I don't know why with my camera is not so clear, but too bright.
Oh, don't worry, we can still see you. Thank you so much.
Yeah. Good afternoon, colleagues. Today, I would like to introduce to you our research with the title Opisthorchis viverrini and a novel sister species in duck share the same intermediate snail and fish host in central Vietnam. Opisthorchis viverrini, a fish-borne liver fluke, the most important public health member of Opisthorchidae, is cause of important morbidity and mortality in humans in lower Mekong region of Southeast Asia. The life cycle of Opisthorchis species involve freshwater snail and fish as the first and second intermediate host. Parasites from the liver deposit embryo eggs, and the egg access on with the feces into environment. Bithynia snail insect the egg hatch inside snail, and after several developmental stages, Saccharia are released and such contact to fish and sitting in the fin, skin, and muscle of the fish become metasaccharia. The final or reservoir host, such as human, dog, cat, ingestion of infected raw or undercooked fish of the metasaccharia acid then mature into adult worm in the liver and complete the life cycle. From the life cycle of fluke, we can see eating raw or undercooked fish that harbor with the viral OV metasaccharia is a key risk factor of the infection. On the other hand, the habitants live nearby water sources, having aquaculture practice, low sanitary by bath routines such as defecating into stream, keep domestic animals freely, thus give the parasites a chance to complete its life cycle. But catch and eating raw or undercooked infected fish are more rich of infection with the parasite. Here you can see Vietnam map with 3 parts of North, Central, and South. Among endemic country of these Opisthorchidae member, Vietnam is unique country have mixed infection. Mixed infection between Chlorochis sinensis and Opisthorchis viverrini. However, Opisthorchis viverrini only found in the central and the south part of the country, and the Chlorochis sinensis found in the north. And predicting that in 2012, our research group discovered a new Opisthorchis species in duck in Opisthorchis viverrini endemic province in central Vietnam. Recording for the first time, 2 Opisthorchis species are coexisting in one endemic area in Vietnam. Here you can see live fish is selling as a local market for live fish. This is the picture on the left hand, and on the picture in the right hand, live raw fish. This is at the mill. The objective of this study to identify which fish and snail intermediate host species play important role in transmission of both Opisthorchis viverrini and Opisthorchis species found in duck. To investigate prevalence and intensity of infection of Opisthorchis viverrini in the intermediate host in an Opisthorchis endemic area in central Vietnam. And more important, to clarify genetic relationship between OV and Opisthorchis species found in duck and other zoonotic members of Opisthorchidae family. A cross-sectional survey was conducted on both snail, fish, intermediate host, and human host to collect data and worm, Metasaccharia, Saccharia, and egg for identification by PCR and sequencing technique. You can see here in the picture on the top, fresh OV were expelled from a patient in central Vietnam. And in the picture in below, Opisthorchis species recover from bioduct of duck in the same study size with OV. And here, list of sample of all developmental stages of Opisthorchis species in duck in Bình province used for molecular analysis in this study, including adult worm, Metasaccharia, Saccharia, and egg. On the other hand, OV samples from this study were analyzed as well. And here, list of primer and target gene for DNA sequencing, including gene code NAT1 and COP1 of mitochondrial protein coding gene, and gene of 18S and 28S of nuclear ribosome gene. For snail hosts, the results show that prevalence of OV in Bithynia snail hosts in Bình province in central Vietnam is a typical low, less than 1%. Among 1,600 Bithynia snail examined, two Bithynia species, Bithynia funiculata and Bithynia siamensis coniumphalus, are infected with OV. The collection size of snail were significantly associated with infection. In that, snail collected from irrigation canal are the most risk with the OV infection. And very interesting that Bithynia funiculata were shared were host with both Opisthorchis viverrini and Opisthorchis species found in duck. For the fish host, total 12 fish species presenting in 6 families were found in this study in the study area of Bình province in central Vietnam. 10 of 12 species in red highlight were recovered with Opisthorchis viverrini Metasaccharia, and the 3 fish species of Esomos metallicus, Puntius brevis, and Raspora aurotenea in red bowl are recovered with both Metasaccharia of Opisthorchis viverrini and Opisthorchis species found in duck. The result, the next result show that prevalence of Opisthorchis viverrini infection in fish is high, up to 74% in Oratus, or zeolfish in local name. A fish normally used for preparation of live fish dish. 3 fish in red highlight are significant associated with OV infection, of which Carassius auratus had the most risk of infection with OV. The intensity of OV infection in fish is low, less than 10 Metasaccharia per fish. This result fit to the low infection intensity in human hosts in the study site. To see the genetic distance between Opisthorchis species and other species, baseline alignment of concatenated COP1 and LAT1 and COP1 nucleotide sequences of Opisthorchis species found in duck were aligned with those of other Opisthorchis species, including OV and CS. The results show that within each Opisthorchis taxon, base-wide genetic distance are small, only range from 0.4 to 0.7% for nucleotide within Opisthorchis viverrini, and for a range from 0.3 and to 0.6 nucleotide within Chloropis sinensis. But this result between Opisthorchis species in duck different from other more than 10%. This means that Opisthorchis species found in duck differ from Opisthorchis viverrini by an interspecies distance. And the same result with base-wide alignment of amino acids. Opisthorchis species found in duck differ from Opisthorchis viverrini by an interspecies distance. This is phylogenetic tree of the combined nucleotide sequences of complete gene COP, NAT1, and COP1 of the mitochondrial gene, so that the Opisthorchis species found in duck clade is blessed as a sister of Opisthorchis viverrini from Thailand, Vietnam, and Laos. And the result of phylogenetic tree of combined nucleotide sequences of nuclear small ribosome subunits 18S and large ribosome subunits 28S show the same result as does the mitochondrial gene. Again, blessed of Opisthorchis species found in duck in a sister position with Opisthorchis viverrini from Thailand and Vietnam. In conclusion, Opisthorchis viverrini and Opisthorchis species found in duck in Bình Định province of central Vietnam are symmetric in Bình Định province in central Vietnam and share snail and fish intermediate host. The symmetric distribution of two related species and the sharing of the same intermediate host may have epidemiological consequence and result in hybridization or introgression among those species. Yeah, and thank you for your attention. And if you have some question, yeah, we can discuss. Yeah, thank you.
Your very informative talk. Are there any questions from the floor? If you have questions, you can ask directly to our speakers or you can put in the chat box. There's a question in the chat box. From the prevalence data, how do you distinguish O. viverrini and O. lobatus metasaccharia from intermediate host fish?
From the snail and fish host, actually for snail it's very difficult. We just pick up some saccharia for DNA from sequencing. But for the metasaccharia in fish, it's easier to differentiate them because the metasaccharia of Opisthorchis species found in duck is smaller. It's much smaller than metasaccharia of OV. But the morphology is quite similar, but the size is smaller.
Oh, the size.
But actually, we didn't estimate the prevalence of saccharia and metasaccharia for each for OV and Opisthorchis found in duck seabirds, we just how can say, when we especially for metasaccharia, as for saccharia in snail, it's very difficult. So we just estimate prevalence when we found with the form of saccharia, the correct form. I don't remember. Bleu, for a long time. I don't remember.
Thank you. There also another question from the chat box. Can you find a hybrid strain of Opisthorchis in Vietnam?
Not yet.
Okay. And another question is that, did you find any mixed infected snail by the two species of liver fluke?
Mixed in, yeah. It's a mixed in. Actually, it's just around 10 infected snail, and we keep we pick up saccharia separate from each snail. Then I pick up some saccharia from each snail, then do PCR with and sequencing. And from the result, we find out that some sequencing, that's the blast, that's identity with the Opisthorchis viverrini, and some sequencing when we blast is identity with the Opisthorchis species found in duck. That is why we make conclusion that it's a share one.
You mean that is from the same snail, right? Is that thank you. So another question from Professor Somchai. Opisthorchis in duck, what is the first and second intermediate host?
It's the same with OV. First intermediate host is the Bithynia snails, and second intermediate host is some some crabfish.